Tech Talk with the technical information scientists at the Jackson Laboratory. My name is Dr. Dolores, and my job is to serve the research community by answering mouse research questions. Today, it's my pleasure to introduce my special guest, Dr. Jim Keck, Senior Director of Innovation and Product Development at the Jackson Laboratory. Great to see you, Jim. Great to have you joining our show.
It's my pleasure, Dolores. Thank you.
So today, let's talk how to test if your drug induces a cytokine storm. In today's episode, we'll discuss why JAKs created this unique platform to help predict CRS, to address the safety problem much earlier in the drug development process, and how you can use this assay to stratify patients as part of enrollments in clinical trials. Some of you may remember our episode number 31, where Xingtong and I went over the basics of what a cytokine release storm is, and that it can be observed in a subset of patients with COVID-19, for example, and also in a subset of cancer patients treated with immunotherapies. These therapies can activate patients' immune system to attack cancer cells, but also in some patients, a cascade immune response also leads to unwanted toxic damage to healthy cells and tissues. So today we will focus on that type of CRS driven by immune effector cells, including T cells and natural killer cells. So basically the way this works is the patient's cells are genetically engineered to express the chimeric antigen receptor in the case of CAR T cells, or bispecific antibodies that are designed to bring together these effector T cells or NK cells in close contact with tumor cells, resulting in increased tumor cell killing. Now, this is a very powerful new types of therapeutics for cancer that they are approved to treat patients' hematologic malignancies. But even though CAR T cells trials have shown very impressive remission rates in patients, they also do have the potential of causing life-threatening toxicities when the patient's immune system starts targeting healthy cells and organs. So, Jim, what are the pros and cons of using immunotherapeutic agents like CAR T cells and bispecific antibodies in the war against cancer?
Good question, Dolores. Immunotherapy, using the human immune system or your own immune system to attack cancer, is an extremely exciting area. The idea of CAR T therapy, we can actually take a patient's own T cells out, modify them, and put them back into the same patient for the purposes of attacking that patient's specific tumor, is extremely potent, powerful technology. Bispecifics, the idea of pulling the immune cells to the cancer cells so they're directly in contact to interact, once again, have a high potency, very exciting areas, and all the related therapies coming out there like CAR-NK and trispecifics. So they're showing preclinically strong efficacy. They're also, what clinical data has been generated with CAR T is extremely promising. The problem we have is that some of these agents, because they're activating the immune system or bringing the immune system closer to cancer, they're causing things to be secreted called cytokines, which are involved in the response to cancer. If the cytokine levels are too high, okay, it leads to toxicities in people. You can see fevers, you can see organ damage, you can see organ failure in very severe cases. So the idea is that while they're very potent, there's significant side effects in some individuals with these therapies. Right.
Thank you. And yeah, I was reading about this, and the rate of CRS associated toxicity it can be pretty high. I don't know if you have numbers. I found some numbers from different papers. I'm not sure. So basically, for example, toxicity can range from 37 to 93% in patients with treated for lymphoma, or 77 to 93% patients with leukemia. So it's pretty high. I was not aware of other ranges of toxicity for other indications? I'm not sure if you have information like that, but it can be pretty high.
It can be very high, and sometimes the cytokine release you're seeing, or toxicity or cytokine storm, is not necessarily related to efficacy. So sometimes you can have these toxicity caused by these cytokines and still not eliminate or reduce tumor burden sufficiently. So it's the side effects are unfortunately something that has to be dealt with, and hopefully we can find ways to predict this going forward in the future.
Right. So, Jim, you come from the drug development world, so you understand that space very well. Why is it that many times drug developers of CAR T cells or bispecific antibodies are caught by surprise in clinical trials when the drugs are dosed in patients? Are they not looking for potential cytokine storms when in preclinical stages? Are they what's going on there?
Sure. Drug developers are looking for efficacy, but they're also trying to find safe drugs, period. The problem is the tools that are used right now are not predictive of what happens in the clinic. In vitro screening of bispecifics or CAR T is in a plastic dish. Only certain components are actually added to this. If you look at triplicates and done in the same experiment, the variability from well to well to well is high. You take the same therapy and give it to multiple labs, you'll get different data from different laboratories. It's high. It's while it's an inexpensive assay, it's not very reliable to predict what's going on in the clinic. A lot of groups also fall back on non-human primates. The problem with non-human primates is obviously if you're looking at an immune modulator, non-human primates don't have tumors. Okay, that's your target. Okay, that's part of the involvement of the cytokine release. You have a monkey immune system, not a human immune system. They don't have cancers, so therefore sometimes it gives you a false sense of cytokine release syndrome toxicity being not present because the targets aren't there. They're good for other toxicities, but not necessarily for CRS. So the in vitro assay is not reliable for what's going on in the clinic. It's highly variable. And non-human primates there are certain components like missing a tumor that's missing that makes it a little less reliable.
Right. Thank you for explaining that in detail. So that brings me to the next question, which is just for you to tell us about the platform that your team developed, and why do you think this platform is more sensitive and more reliable for detecting human CRS compared to the assays or the methods that you just described?
Thank you, Dolores. It's a great question. So about 5 years ago, here at the Jackson Laboratory, we wanted to develop a platform that would be able to better mimic what's going on in the clinic if possible. So we've developed using the NSG mouse, which is the immunodeficient mouse we have at the Jackson Laboratory, but also variations of that, like the NSG SGM3 or MHC class 1, class 2 knockout, as well as other strains. We took them and we humanized them with PBMCs from normal volunteers. We used leukopaks, which is essentially a large volume of these PBMCs from individuals.
So they're human PBMCs, right?
So absolutely, Doris, you're human. So we humanized the mice with the human immune system using these PBMCs. And then what we do is, because it's got all the endothelial cells present, it's got the human immune cells in there, we can actually introduce human tumors as well. And what we found is that we developed an assay that's fast, sensitive, reliable, and highly reproducible, that we could actually look at immune modulators, CAR Ts, bispecifics, T-cell engagers, in the presence or absence of tumor, but in the presence of the immune system and the target for the therapy. We can actually look at things like cytokine release syndrome, but in the context of a tumor. So it allows us to be able to do cytokine release syndrome and efficacy in the same mouse at the same time.
Perfect. That sounds great. So for those of you who are interested, we have a lot of data. Jim has a lot of data available, Some of that you can see in our on-demand webinar presentations that contain detailed characterization of this very cool platform to evaluate more accurately and the safety and the systemic toxicity of potential immunomodulatory drugs in a more reliable way than in vitro. So feel free to contact us for more information regarding this platform. If you see the webinar and you're more interested, we definitely would like to talk to you. So, Jim, the next question is, can you explain how a drug developer can use this platform in their evaluation of new therapeutics earlier in the development pipeline?
Sure, Dolores. A major goal of this assay was to de-risk drug development. Okay, because we can basically have a single mouse that we can actually put in the tumor with the human PBMCs. We can look at efficacy, cytokine release, the toxicity on the mouse, changes in the immune system, and downstream organ damage in some cases, all in one mouse in groups of 4 or 5 mice per arm because of the tightness of the data. It gives you a chance to look at your drug development in the context of many different variables all at the same time. So if you're going to be starting screening a new antibody target and you develop a series of like 20 different antibodies, you could actually from the day one start asking questions about efficacy, but in the context of safety and effects on the immune system with that drug. So it gives you a better chance to capture both at the same time. Most drug developers look at efficacy first, followed by toxicity, but we can do both in the same mouse in the context of each other. So it makes it more efficient and gives you more information. But what we also can do is dose ranging. We can actually increase the dose of the various therapies and look at the effects on efficacy and toxicity to find a better dose that's going to give you a maximum efficacy with minimal toxicity. And then you're allowed to look at a population of individuals, not just one PBMC donor, but 10 or 20 if you choose to see how a population will respond to your therapy as well.
Right. That sounds fantastic. Right. No drug developer wants to spend money, time, and effort developing a drug that is going to be too toxic by the time the patients get it. That's really an important thing that people have in mind, and it's better to kill that candidate or to select a better one earlier in the process and not by the time you're in clinical trials.
Correct.
So next question, what could drug developers do differently if they knew that the drug may have adverse immune side effects as the drug starts being enrolled in clinical trials? So let's say that the drug already made it to that point of clinical trials. You're enrolling patients. I mean, how can your platform inform or better inform the patients that you're going to enroll in those clinical trials?
Another good question, Dolores. So in theory, what we're trying to accomplish here is a way for you, the drug developer, to better understand how your drug's going to perform in the clinic. We've de-risked it preclinically, but now we're looking at going into phase 1 that's safe for safety. Because we can use PBMCs from normals and probably from patients, we can actually humanize the mice and look at a population response and how a phase 1 in this model could possibly give you some insights on how 20 different individuals will respond to this therapy. Whether it's a normal PBMCs or patient PBMCs allows you to do that. It gives you a chance to better understand going in, you're gonna have issues going forward. It also allows you to be able to examine sensitivity of these people. So let's say you have a patient population of 20 people going on trial. Some people are going to be extremely sensitive to this drug. So you're not going to be able to dose it high enough to get efficacy. Maybe these individuals should be getting a different drug. Meanwhile, the other people that are less sensitive to the toxicity, you could best keep them on trial and actually raise them the dose to get to a higher efficacious dose possibly, because they're less sensitive to CRS. Or if you wanted to keep everybody on studies, if you know ahead of time who's going to have a cytokine release syndrome or a storm, you could actually prepare to pre-treat these patients to diminish the toxicity while on trial. It gives you more insight about potential, but also gives you potentially more insight and ways to use the information to further develop your drug.
Right, great. So while you were saying that, I was thinking, I mean, this platform may also be interesting and a valuable thing to have for those centers that are actually administering this type of drugs, because the centers that are administering this type of drugs, they have to be certified transplant centers for now, but the thing is they don't know ahead of time who's going to respond badly and who's going to respond fairly well. So it would be giving them heads up on how involved the management of short-term or long-term toxicities may be, right?
Absolutely. One thing I hear from CAR T or cell-based therapy physicians is that it'd be good to know ahead of time who's going to get a severe response or severe cytokine storm. And by knowing so, they can actually be prepared for them to be able to treat those patients when they're on therapy. But in the case of things like allogeneic or off-the-shelf CAR T, it's almost like going to be more of almost like a classical clinical trial, in that you're using the same therapy for a large population of individuals,
right,
off the shelf, to be able to actually understand early on who's going to get this toxicity and who's not, who's going to get efficacy, who's not, would be great information for a developer as well.
Right, great. So the assay sounds very, very exciting, and I am looking forward to seeing more data that demonstrates the clinical relevance of it. So can you tell us more about the collaborations that you are seeking towards demonstrating this goal?
Yes, Dolores, this is a key point that we want to emphasize here, is that preclinically this model is been done hundreds of times. We have a lot of people utilizing it, but we have yet to demonstrate fully clinical relevance of this platform. We're in a process of setting up collaborations to better understand how this platform will be predictive of what happens in the clinic. So we're looking for collaborators to work with us on bispecifics, possibly CAR T as well, to see if we can do retrospective analysis of trials. Give an example, if you finished a phase 1, you had some patients with CRS, patients without. Could our assay have predicted it up front? Working with us, we can actually look at those patients' PBMCs to understand if this assay would have been predictive or not. It would be very useful for the drug discovery world, as also possibly patients in the future.
Absolutely. That reminds me of what it took to show that patient-derived xenografts PDX were a useful tool to replicate patients' clinical outcomes. At the beginning, it was just a thought, and then nowadays it's a well-known fact that most of the times PDXs, so basically patient-derived tumors engrafted into immune-deficient mice, actually the same mice that you're using for this platform, they, if you treat the mice with certain drugs, actually it's predictive, has a predictive value if you compare it to the clinical outcomes. So it did remind me of that stage of the PDX development as well.
In having collaborators to help us validate these types of platforms is critical to Jackson Laboratory, and we encourage people to contact us about ways we can work together to help find better drugs. Great.
Yeah, that's definitely part of our mission to improve human health and to support researchers around the world. So it's right on our mission there. So this is all the time we have for questions this week. If we didn't answer a question, let us know. You can email it at mystech@jax.org, and we will answer it for you. I wanted to mention that we do have several links at the bottom of the video description. The first one is a short video describing what CRS is. The second link is a link to the JAK Cytokine Release Syndrome evaluation study page to look at the safety of your therapeutic prior to entering clinical trials. The third link is a webinar where Dr. Jim Keck describes in much more detail the new humanized model for prediction of CRS risk in vivo. So thank you so much, Jim, for sharing your development with our viewers. I really appreciate that. And our next MyStech Talk is episode number 38, called Let's Talk Pharmacokinetics for Antibody Development, next Tuesday, June 15th. We look forward to seeing you in LinkedIn or YouTube. And lastly, I wanted to mention that over the next few weeks, we will be transitioning the name MyStech Talks that you got used to be called JAK's Tech Talks. And this is a new name that reflects a future of on-point tech talks discussing a variety of thought-provoking and educational topics, including novel experimental platforms, as well as innovative approaches for discovery research and practical advice to support successful use of mouse models in preclinical and basic research. So stay healthy, stay safe, and stay excited about your research, and see you next time.